Welcome back to Falcon Physician Review's online review for USMLE Step 1. This is Microbiology, Module 27. We're going to talk about viral assembly and release. In the last module we talked about viral replication and how each virus gains its DNA and gets it to make proteins so they can release. That's what we'll talk about in this module, how different enveloped and non-enveloped viruses get out of the cell and go on to further the infection or infect other people. Welcome to Falcon Physician Review's online review for USMLE Step 1. This is Microbiology Module 27, Viral Assembly and Release. Assembly and release differs between naked and enveloped and complex viruses. A naked virus has maturation, which consists of two main processes, which is assembly of the capsid, which is usually done by late proteins, and association with nucleic acid, which is synthesis. Maturation occurs at the site of the nucleic acid replication, regardless of what's going on. The structural proteins in genome, RNA or DNA, assemble into the intact helical or icosahedral virion, and then it's released. For enveloped viruses, it's a little bit different. The capsid is assembled around the nucleic acid to form the nucleocapsid. The virus-encoded proteins then migrate to the plasma membrane or the nuclear membrane. Envelopes are formed around the nucleocapsid by budding of cellular membranes. So this way, envelope viruses acquire the host membrane that they had where they infected the cell. For complex viruses, like smallpox, molluscum contagiosum virus, assembly occurs in factories in the cytoplasm. They acquire a membrane while still in the cytoplasm, and they're released by host cell disintegration. Naked viruses, the cell may lyse and release the virions, or they may be released by reverse phagocytosis or exocytosis. This figure shows the life cycle of an enveloped RNA DNA virus. It looks like an HIV virus because it requires reverse transcriptase. Up in the top right we have binding, where the membranes on the envelope target the host cell tissue. Then you've got fusion into the cell, where the capsid is uncoated and it's there in the cytoplasm and reverse transcription takes place. Then you have nuclear localizing, where the RNA and the DNA go into and can integrate into the host DNA. If you have a latent infection, it can stop here for months or years. Eventually, though, however, the genes will be transcribed and translated, and then you'll get genomic RNA out into the cytoplasm, where you make the proteins that are needed for the virus to assemble, to bud, and to release. That's what you see on the bottom right. So that's how a virus goes from start to finish, using the host to accomplish its designs. Let's do some questions. Which of the following statements regarding replication in retroviruses is true? A. The reverse strand is transcribed to complementary DNA by the virus-associated reverse transcriptase. B. Complementary DNA is transcribed to messenger RNA using host RNA polymerase. C. Both of the above, or D. None of the above. We have to remember that retroviruses are RNA viruses, and they need to turn into DNA viruses. And so the reverse strand is transcribed to complementary DNA by the virus reverse transcriptase. So A is true. But then the complementary DNA can be transcribed to messenger RNA using the host RNA polymerase. So both are true. The answer is C. Next question. During the process of DNA virus replication, the early genes encode for which of the following structures? A. Enzymes and regulatory proteins. B. Structural proteins. C. Both, or D. None. So we know that early genes encode for enzymes and regulatory proteins needed for viral replication. The structural proteins are usually more indicative of the late genes. During the process of DNA virus replication, which of the genes encode for the structural proteins needed to assemble the mature virus? Well, we just answered that. It's B, the late genes. During DNA viral replication, the capsid structural proteins are synthesized from. So we have capsid structural proteins. These are associated for the late phase. They're what you need to complete the virus so it can bud out, and so it's going to be a late messenger RNA. Next question. Which of the following is true regarding the replicative form during viral RNA synthesis? So the replicative form includes two double strands, a positive and a negative strand. And that's going to be A. It's not partially double-stranded. It does have the two strands, one positive and one negative, and that's the replicative form. You can have a replicative intermediate, but that's not going to have the two strands. Next question. A virus was isolated from stool, and its genome was sufficient to initiate virus replication upon microinjections into cells. So just from thinking about that, we know that if we found it in the stool, it's not going to be enveloped, okay? It has to be a naked DNA, and so we only have to now worry about whether it's DNA or double-stranded RNA. The fact that it was able to replicate upon microinjection into cells means that if we just take its genome, it is able to make proteins. In order to do that, we have to have either positive sense RNA or DNA, and so D is the correct answer. Double-stranded RNA doesn't have the viral RNA-dependent RNA polymerase in order to make positive sense RNA. Next question. During the process of viral replication, the stage between the end of uncoating and maturation of new viral particles is known as. This is a name that you have to just have down cold. It's the eclipse phase. It's after uncoating, and it's before you find new viral particles, where you can't actually find any complete virions within the cell. So if you have a host cell that's been infected, you only have a few options open to you. One is death by lysis. One is transformation, where they activate or induce oncogenes, resulting in uncontrolled or uninhibited cell growth. Examples of this include cervical cancer from HPV, Burkitt's lymphoma from EBV, hepatocellular carcinoma from HBV. You can also have latent infection or chronic slow infection with herpes virus or hepatitis B and C. Our bodies do have immune responses to viruses, and antibodies are important. They can kill the virus before it gets into the cell. Once it's in the cell, we have cell-mediated immunity, which is necessary for enveloped or not lytic cells, lytic viruses. We have protective antibody directed at viral attachment proteins, and cell-mediated immunity directs towards peptides from any protein found in the virus. Wrapping up Module 27, it's fairly simple. If you're an enveloped virus, you have to bud through the host membrane. You acquire host membrane proteins, and you're dependent on the host being alive and intact to use its membrane in order to get out. Then you'll use target tissues and enzyme receptors to get to your new surface and be able to form an infection there. If you're a non-enveloped virus, you can just kill the cell and you don't require its membrane in order to move on. These two strategies are important, and it's a fundamental difference between enveloped and non-enveloped viruses. Now that we've talked about the viral life cycle, we're going to talk in Module 28 on the different agents we have developed in order to kill viruses.